DNA repair by direct reversal in eukaryotic systems. So this session is going to be an overview of the most prevalent direct reversal DNA repair mechanisms. So let us look at the learning outcomes of this session. In eukaryotes, direct reversal of DNA damage is mainly for alkylated products formed by alkylating agents. There are two enzymes that carry out direct reversal. One is the O6-methylguanine DNA methyltransferases, commonly called as MGMTs, and alpha ketoglutarate based hydrogenases or dioxygenases, which belong to the alpha Kg Fe dioxygenases. Now, the MGMT has several conserved domains, and its action is considered to be a suicidal mechanism, which means that once it carries out the reaction only once, and after that it cannot carry out a second reaction. It is also a housekeeping gene, which means that it is found in all cells. ALKBH is reported to reverse alkylated adducts by the mechanism of oxidative demethylation. So let us first just look at getting an overview of what are the alkylating agents and the alkylated adducts formed. We are not going to go into too many details, but just an overview. So alkylating agents can be from exogenous sources. So for example, the environment, you can get it from air, water, food. For example, nitrogen mustard is one of the alkylating agents, and most of the alkylating agents are, say for example, nitrosoguanidine derivatives, nitrosourea derivatives, and methyl sulfonates. A lot of studies have been done with ethane methyl sulfonates, and these have shown to be able to induce a lot of mutations. So a lot of mutagenetic studies are being carried out by using EMS. Now you also have endogenous agents. Now these are products that are either byproducts or end products of several biochemical pathways. For example, S-adenosyl methionine can contribute to methylation. Nitrosation reactions can lead to methylation, and so both are able to, or both these type of agents, whether exogenous or endogenous, are able to cause methyl adducts both in the DNA and in the RNA. And the methylation is generally either O alkylation or N alkylation, which means that both O and N, whether it is exocyclic or whether it is part of the aromatic ring itself, these N and O's can be alkylated. And on alkylation of these nitrogen bases, what happens is that many a times these nitrogen bases are not able to form a proper interaction with the complementary nucleotide, and hence there would be some sort of distortion in the DNA. This distortion is or can disrupt several molecular mechanisms, such as it can stall a replication fork, so replication can be disrupted, transcription can be disrupted, etc. Now, because it affects the basic molecular mechanisms of the central dogma, one observes that if there are a lot of alkylations happening, and therefore the frequency of mutation is increasing, then it leads to what is called as diseases like cancers, etc. Now, having looked at them being the alkylating agents, let us just look at which are the major sites in which the alkylation is greater. So, the N over here, the O, N, and the N, which are part of the cycles, are the major sites where alkylation is possible, and these are the ones where minor damages are possible. Similarly, here you can look at where you have minor damages and where you have the major damages. Moreover, what is also observed is even the phosphates, the phosphodiester bond can be methylated, and that too can prove to be detrimental in many cases. So, what has been studied and reported is that O alkylations are highly mutagenic, N alkylations are less mutagenic, but they can be cytotoxic. And the products of the alkylating agents are 1-methylguanine, O6-methylguanine, 7-methylguanine, 3-methylguanine, 3-methylcytosine, 3-methyladenine, and many more. So this was just an overview on alkylating agents and alkylated adducts. Now we go to the methyltransferases. In mammalian systems, mainly the 1-methyltransferase that is most prevalent is the O6-methylguanine DNA methyltransferases, and therefore the MGMT that is found even in humans is mainly of the O6-methylguanine methyltransferase. Now, to just look at the general mechanism of how methyltransferases function, just before that, let's look at the fact that suppose you have the guanine alkylated, so you have a methyl group that is added at the sixth position, and therefore this is called as the O6-methylguanine. Now, O6-methylguanine, where the guanine should have actually complementarily bound to cytosine, and O6-methylguanine binds preferably to thymine. So if this interaction that is happening because of O6 methylation or the adduct being present, if the replication is going forward and the thymine is added, then what we will observe is that a GC base pair will have converted to an AT base pair, and that definitely is a mutation. So what is observed is that this OCH3 that is present is bound to by the methyltransferases. It is ensured that it is present within the active site of the methyltransferases, and the methyl from the nitrogenous base is actually transferred to the cytosine. There is one conserved cytosine within the active site of the methyltransferase. So the CH3 from this O is transferred to the C of the methyltransferase, and then having done that it leaves. So therefore what is observed is that the CH3 is in a stable bond with the cysteine of the methyltransferase. Now of course, what is restored is the normal base, but then what is the end result of this reaction is that the methyltransferase, once bound to the methyl, is now no longer able to act as an enzyme, and therefore this reaction that it carries out makes it inactive as an enzyme further, and hence such enzymes are called as the suicide enzymes. Now, what is observed is that this mechanism that one observes, this general mechanism that one observes, is quite conserved across species. And this methylated methyltransferase is then ubiquitinated and degraded by the proteasomes. So therefore that is very interesting that methyltransferases, they become inactive, but they are saving the DNA from the results or the consequences of the methylation, and that is how this enzyme is sacrificing itself for the genomic stability to be restored. Now, let us look at just the structure of the methyltransferases. As mentioned earlier, it is a monomer, but it has several important domains present in the monomer. One important C-terminal motif that is present is this helix turn helix motif. It also has zinc finger. It also has the conserved cysteine 145, and it has the beta sheets present at the end terminal. Now, together all of these enable binding to the DNA. So you can see that you have the helix turn helix binding specifically to the DNA, and the recognition helix binding to the methylated adduct. Now, interestingly, this helix turn helix has an arginine finger which can flip out the target nucleotide. So, the methyl guanine is flipped out, and after it is flipped out, at the catalytic site of the methyltransferase, the methyl from the guanine is transferred to the cysteine. So, here's the cysteine. Now, the methyl is transferred to the cysteine. So, thereby, you have the methyl permanently bound to the cysteine residue of this. Okay, so the next set of enzymes that is observed to be carrying out direct reversal is belonging to a ketoglutarate dependent dioxygenase family. And these group of, so it's not that all, so this belongs to this super family of dioxygenases, which are Fe2 ketoglutarate dependent dioxygenases, But not all ALKBH are responsible for DNA repair. In humans, it is found that ALKBH 1 to 3 is responsible for direct repair, and in other species, there are other ALKBHs which are responsible for direct reversal of again alkylated adducts. And they have observed to be commonly reversing 1-methyl adenine and 3-methyl cytosine, although it can also reverse 3-methyl guanine, 3-methyl thymine, etc. 1-methyl guanine, all that is also something that is reversed by ALKBH. The process or the mechanism by which it is able to reverse these adducts are through a process that is called as oxidative demethylation. As you can see over here, most of the methylated nitrogen bases or even cyclized adducts that are there, they are basically converted in such a form that they react with the molecular oxygen, and after reacting with the molecular oxygen, the alpha Kg, which is a coarse substrate, gets decarboxylated and forms succinate, and the carbon dioxide is released. So this is part of the reaction through which it is carrying out demethylation. It forms several intermediates, after which one gets what is called as a hydroxylated methyl at the position that it was in, and this hydroxylated methyl is then removed in one or other hydroxyl methylated product. So here you can see that this is a formic acid that is being for formic acid that is being removed. Okay, sorry, formaldehyde that is being removed. I'm sorry. So thereby you can have different hydroxymethylated groups that can be removed, but eventually what you will get back is the original base, and that is extremely important. But in the process of carrying out this entire thing, one needs alpha Kg, one needs the Fe in its active site, and therefore it is basically an Fe2 ketoglutarate dependent dioxygenase. Now, looking at its structure, again, what one finds is that there are several conserved domains in the ALKBH. So you can look at different ALKBH and you will find a structure which is very similar. Plus, you will find that in its active site is present Fe2 plus. Whether it is ALKBH3 or it is ALKBH2, you will find the same Fe2 plus present in the active site. That is something that is very common to all of them. Now, what happens is that within the active site domain is present a very special motif which is called as histidine X aspartate, okay, or glutamine X again histidine. So this motif that is there plays an important role in enabling the reaction to happen. So the damaged place, again in this case, therefore, the different motifs that are present are able to flip out the active site. In case of ALKBH2, it has been observed that there is a tyrosine residue that can hydrogen bond with the phosphates around the methyl group, thereby positioning the methyl group at the active site. And because it is able to position the methyl group at the active site, it is able to hydroxylate the methyl group. Now, the active site which is present, which has an Fe2 plus, that Fe2 plus is able to bind to molecular oxygen to form what is called as an oxo intermediate. And that oxo intermediate reacts with the alpha ketocarbon of the alpha ketoglutarate to form another intermediate, and by in lieu of forming another intermediate, it releases succinate and carbon dioxide. So thereafter, another intermediate over here is formed, and from that intermediate, one gets a methyl group which is hydroxylated. So the final product that is released is a hydroxylated methyl group. And therefore, what is important to note is that the methyl group doesn't remain attached to the active site of the enzyme, it is released. And hence, this enzyme is not a suicide enzyme. The enzyme can go back and bind to another methyl adduct, remove the methyl, and correct it. So therefore, ALKBH can do several rounds of DNA repair. Now let us therefore go to the conclusions. The MGMT, especially the O6 methylguanine methyltransferase acts as a suicide enzyme to remove a methyl group from a methyl adduct. The MGMT has a mechanism of flipping the methyl adduct into its active site and transferring the methyl group to a cysteine residue within the active site. The ALKBH proteins are able to repair the damaged methyl adducts in the DNA by oxidative demethylation using molecular oxygen, alpha Kg, and the methyl group that is released is finally removed as a hydroxylated methyl group. The ALKBH group of enzymes too are able to flip the methyl adduct into its active site, which has an Fe2 that can react with molecular oxygen to carry out demethylation. Thus, direct reversal of DNA damages are the simplest repair systems that are carried out to minimize the eventualities of mutation and cell death. Several studies have been carried out where mutations in MGMT and LKBH have shown to give rise to numerous or high frequency mutations, and that has led to genomic instability. Thank you.